Hi there everyone, thanks for tuning in again. This is Sir ML. In this video, I will just show you how to do a simple monohybrid cross using the classical example that was first cited by the myth, the legend, the man himself, Johann Gregor Mendel, who was considered to be the father of genetics. He was not only that, but he was a teacher, he was a botanist, he was a mathematician as well as an Augustinian friar. So he was definitely a man of many talents and definitely a legend in the world of genetics. In this cross, we will be doing a simple mathematical deduction to see how a gene that codes for a single characteristic or trait, in this case the stem height of the pea plant, is being inherited through generations. So let's start by crossing a tall pea plant and a short or dwarf pea plant. The tall pea plant and the short pea plant here will be the first plants to be crossed for this particular monohybrid cross. So we can consider them to be the parents or belonging to the parental generation. So we can assign them with the capital letter P, which means they belong to the parental generation. We can assume that both of these plants are homozygous for their condition, and we can represent this by denoting the alleles using specific letters. In this case, we can use the first letter of the dominant phenotype or dominant trait, which is T. So for the tall pea plant, the genotype can be represented by two uppercase letters T, and we can use the lowercase letters t for the recessive short plant. Now, one thing to know is that before they can pass on their traits and their alleles to the next generation, obviously they have to produce gametes, right? So they will go through the process of gametogenesis. Now this particular gene which codes for the stem height of the plant, what will happen is it will also be passed on to the next generation, and the tall pea plant will pass on the alleles that we see here to the next generation, while the short pea plant will also pass on its alleles to the next generation. Now when they undergo the process of gametogenesis, remember that in gametogenesis there will be a reduction in the chromosome number, so everything will be reduced by half. The next step here is that the alleles for the tall pea plant, one capital T will be 1/2, right, and the other capital T will be another half, okay, and these will segregate during the process of gametogenesis according to the law of segregation that was obviously given by Mendel. Similarly, with the short pea plant, the two alleles here will segregate into different gametes like that. And so now what will happen in the next step? Fertilization, right? Those gametes of the tall pea plant and the gametes of the short pea plant will be fused to form another individual. So let's see what will happen here. We can do this by using what we call as a punnett square, okay, and here we can count the number of alleles here: 1 2 3 4 alleles. So 2 alleles from the tall pea plant and 2 alleles from the short pea plant. So 2 by 2 will be 4 squares. So we can draw punnett square like this that contains 4 squares, and on one side of the punnett square we can keep the alleles of one of the parents, okay, and in the other side we can keep the alleles of the gametes of the other parent of the recessive parent. Okay, so now what we can do is just simply fill in these boxes, these squares here, by the alleles that correspond to the correct square here. So you can see here in the first square, capital T and small t. Second square, that's the same thing. Third square, similar thing, and even in the fourth square. So we can see here that in the next generation, which we will refer to as the F1 or the first filial generation, we can see that all plants are having both dominant and recessive alleles for stem height. One thing that we know for sure is that the dominant allele will always be expressed over the recessive allele for this simple monohybrid cross. So it means that all the F1 plants will be tall plants. All of them will be tall plants, even though one of the parents was a short plant. Okay, so all the plants are tall. But what is one thing that we notice here is that the genotypes or the genetic makeup of these F1 plants for the particular gene that codes for stem height, all of them are heterozygous. They carry both the dominant allele as well as the recessive allele. So they are heterozygous. So this was one of the results that Mendel acquired when he crossed a tall pea plant and a short pea plant. He found that in the next generation all plants were tall. There were no short plants. And to find out why this has happened, he crossed two of these F1 generation pea plants, and both of them are heterozygous pea plants. Okay. Of course, he didn't know this at the time. What he did was he just simply crossed the tall pea plants that he got in the F1 generation. Now we know that, this is what happens, right? We know that the F1 plants are all heterozygous because we knew the genotypes of the parents, but Mendel did not know this. So what he did was simply cross them and analyze the results later on. But easy for us, we know what the genotypes are. So what happens when we cross these two F1 plants? So again, before they can pass on their alleles and genes to the next generation, they have to go through the process of gametogenesis. Okay, so again, for the first plant, there are two alleles. Okay, this capital T will be 1/2 of the gene, and the small t will be 1/2 of the gene. Similarly with the other plant as well, the gametes that are produced are big T and small t. Both of them will represent 1/2 of the complete set. So again, we can use a punnett square here. Through the square, again there are two alleles from the other parent and two alleles from the other parent. On one side we can put down the alleles of one parent, and on the other side we can put down the alleles on the other side. So let's see what happens when we fill in those boxes. In the first box we have a homozygous tall plant. So again, we can see the reappearance of one of the genotypes of the parental generation. Second box, we have a heterozygous tall plant. Third box, we also have a heterozygous tall plant. And in the final box, in the fourth final square, we have the reappearance of the recessive parent, right? Homozygous recessive parent. So these will be the F2 generation or second filial generation. So let's see. So when we look at the physical appearance of these plants, 3 out of 4 plants are tall and 1 out of 4 plants is short. So that will be the phenotypic ratio, 3:1. But when we look at the genotypes of these plants, okay, the first plant is homozygous dominant. 2 out of 4 plants are heterozygous dominant, and 1 out of 4 plants is homozygous recessive. So what do we have here? We have 1:2:1. So that will be your genotypic ratio. Hope you found this video helpful, and if you do, please go ahead and hit that like button. Thank you.